Welcome to the Weekly Neurology Deep Dive, produced by Gavanini. Today we're taking a deep dive into some really intriguing research. It connects medications we typically associate with human immunodeficiency virus and hepatitis B to potentially a reduced risk of Alzheimer's disease.
It is a compelling study, and just to be clear up front, as always, this deep dive is purely for educational purposes. We don't have any commercial goals here. What we're doing is summarizing the research paper, and we really do recommend you dive into the full publication yourself for all the nuances and details.
Absolutely. Let's start perhaps with a quick practical summary for listeners who might be pressed for time.
Good idea. So the main takeaway is this. The study found that exposure to a specific class of antivirals, known as nucleoside reverse transcriptase inhibitors, was linked to a significantly lower risk of developing Alzheimer's disease. And this wasn't just a small observation, it showed up consistently in two very large, quite diverse health insurance databases. The proposed reason: it seems these drugs can inhibit something called inflammasome activation, that's part of the inflammatory process, and this action is separate from their antiviral job. What's also interesting is that other types of antiretrovirals, like non-nucleoside reverse transcriptase inhibitors or protease inhibitors, didn't show this protective link. So the findings really build a case for looking more closely at inflammasome inhibitors as a potential strategy for Alzheimer's.
That's a fantastic summary. It really sets the stage for why this is catching attention. Alzheimer's is, as we know, a huge global challenge, and the search for effective treatments is relentless. This focus on neuroinflammation, it feels like a really important emerging angle.
It absolutely is, and this study specifically zeroes in on these nucleoside reverse transcriptase inhibitors, which are already FDA approved, already out there. It explores their potential in Alzheimer's, not through their antiviral effect, but through this inflammasome inhibition. It kind of challenges, the traditional view that's so heavily focused on amyloid beta and tau. It suggests inflammation itself could be a key target.
Okay, let's dig into that biological concept then, the inflammasome. Can you unpack its role in Alzheimer's pathology a bit more?
Right. So think of the inflammasome, specifically the NLRP3 inflammasome in this context, as a sort of danger sensor within brain immune cells. The current thinking in Alzheimer's is that amyloid beta aggregates and those hyperphosphorylated tau tangles, they don't just cause direct harm, they also trigger neuroinflammation. The NLRP3 inflammasome is believed to be a key mediator here. It senses these abnormal protein accumulations. When it gets activated, it sets off a chain reaction. You get caspase-1 activation, release of inflammatory signals like interleukin-1, and ultimately this contributes to neuronal damage and death. It can even create a kind of damaging feedback loop, worsening the disease. Now, the crucial link for this study is that earlier research had shown NRTIs can actually inhibit this inflammasome activation. And that insight that these drugs had this sort of side effect was the foundation for their hypothesis.
That makes sense. So the biological plausibility was there. How did the researchers then go about testing this hypothesis using real-world patient data? What was the study setup?
They used a drug disease cohort study design. Essentially, they looked back at health records, and they used two absolutely massive data sources. First, the U.S. Veterans Health Administration database that's huge, covering about 11 million people over 24 years, from 2000 to 2024. Second, the MarketScan database, which includes around 158 million people with employer-based insurance over 14 years, 2006 to 2020. So really extensive data.
Incredible scale. And who specifically were they looking at within those millions of records?
They focused on patients who had a record of either human immunodeficiency virus or hepatitis B. They needed to be at least 50 years old when their condition was first noted in the records. And critically, they couldn't have an Alzheimer's diagnosis before that point. Exposure to nucleoside reverse transcriptase inhibitors was counted if they had at least one prescription filled, and they tracked how long cumulatively people were exposed over time. The outcome they were measuring was a new diagnosis of Alzheimer's disease appearing in the record.
Now, with observational data like this, dealing with confounding factors, other things that could influence the outcome, is always a major challenge. How did they try to ensure the comparison was fair?
They were very thorough on the statistical side. They adjusted their analyses for a lot of baseline factors, things like age, sex, race, where available in the VA data, a general comorbidity score called the Charlson score, and also around 20 specific conditions known to be linked to Alzheimer's risk, like high cholesterol, type 2 diabetes, hypertension, history of traumatic brain injury, Parkinson's disease, and so on. Beyond just adjustment, they used propensity score matching. This is a technique to try and create groups of patients, those who took NRTIs and those who didn't, that looked as similar as possible at the start, minimizing selection bias. In the VA data, this resulted in matched groups of about 7,600 patients each, with good balance on characteristics. In MarketScan, the matched groups were larger, nearly 22,000 patients each, also well balanced. And one more layer, specifically for the VA data, they used a fine and gray competing risk model. This accounts for the fact that in populations with chronic illnesses, some people might unfortunately pass away before they ever get an Alzheimer's diagnosis. This model helps ensure the effect they're seeing is truly on Alzheimer's risk, not just skewed by differences in lifespan.
Okay, a very robust approach then.
Yeah.
So after all that careful analysis of these huge data sets, what did the results actually show? What was the impact on Alzheimer's incidence?
The results were pretty striking and consistent across both data sets. If you look at the Kaplan-Meier curves, which track diagnoses over time, there was a significantly lower incidence of Alzheimer's disease in the groups exposed to NRTIs. The log-rank tests comparing the groups were highly significant in both databases, p-values less than 0.01. So a very low probability this difference was just random chance.
Can you give us the numbers? How much lower was the risk quantitatively?
Yes. So looking at the hazard ratios, which estimate the relative risk in the original unmatched VA cohort, each extra year someone was on NRTIs was associated with about a 4% lower hazard of getting Alzheimer's. In the original MarketScan cohort, that was even stronger, about a 10.3% lower hazard per year of use. Now, after they did the propensity score matching and adjusted for all those confounders we talked about, the protective association actually became more pronounced. In the matched VA cohort, it was a 6% lower hazard per year of use. And in the matched MarketScan cohort, it was a 13% lower hazard per year of exposure.
And that competing risk analysis you mentioned for the VA data, how did that affect the numbers?
Right, when they factored in mortality as a competing risk in the VA data, the protective effect was even larger. NRTI exposure was linked to a 32% lower hazard of Alzheimer's in the original cohort and a 37% lower hazard in the matched cohort when accounting for death. This really reinforces the idea that the NRTIs were specifically reducing Alzheimer's risk, not just affecting overall survival in a way that masked the outcome.
Was this effect seen across the board in both the human immunodeficiency virus and hepatitis B patients?
Yes, they did subgroup analyses. The protective association held up for both patients with human immunodeficiency virus and those with hepatitis B, with statistically significant results, p-values below 0.05 in all cases. This suggests the effect isn't just tied to one specific underlying condition, and, really importantly, they checked other classes of anti-HIV drugs. Exposure to non-nucleoside reverse transcriptase inhibitors or protease inhibitors or integrase inhibitors, none of those were linked to a lower Alzheimer's risk. That specificity really bolsters the hypothesis that it's the inflammasome inhibition, which is characteristic of the NRTIs but not those other classes, that's likely driving this protective effect.
That is powerful data. So bringing this back to clinical practice and future research, what are the implications for neurologists thinking about Alzheimer's disease?
Well, I think the consistency across these large, distinct databases really strengthens the argument for the inflammasome pathway being a legitimate target in Alzheimer's. And the finding related to NRTI specifically suggests these drugs, or similar ones acting on that pathway, could offer neuroprotection. The concept of repurposing drugs that are already approved and well understood is, of course, very appealing. It could potentially speed up the development process, which is desperately needed for Alzheimer's.
Of course, we always have to consider the limitations, especially with claims data studies. What did the authors acknowledge as caveats that we should keep in mind?
They were quite clear about the limitations. Claims data, for instance, doesn't usually contain genetic information like APOE status, which we know influences Alzheimer's risk. It also doesn't allow for tracking disease progression with detailed cognitive tests or functional scales. You basically just know if a diagnosis code appeared or not. And there could be unmeasured differences in things like healthcare access, socioeconomic status, or even how readily doctors diagnose Alzheimer's between the different populations or healthcare systems. These are the kinds of things that only a well-designed randomized controlled trial can truly address.
Understood. So given those limitations, but also the promising signal, where does this leave us? Are there trials happening now? And what about safety if these drugs were ever considered for Alzheimer's specifically?
Yes, there is ongoing work. Lamivudine, one specific NRTI, actually just completed a pilot clinical trial in Alzheimer's disease. The report suggests it showed some positive effects on biomarkers related to neurodegeneration and neuroinflammation, which is encouraging. And emtricitabine, another NRTI, is currently being tested in an ongoing trial. So we're starting to see prospective human studies. Now, regarding safety, for neurologists to consider NRTIs are FDA approved, but they do carry known risks, primarily mitochondrial toxicity. This happens because they can interfere slightly with our own DNA polymerases. This can rarely lead to serious issues like lactic acidosis. It's important to stress, though, that the newer generation NRTIs, like lamivudine, have a much lower risk of this compared to the older drugs in the class. In the context of HIV or hepatitis B treatment, this risk is managed with regular lab monitoring, typically twice a year. Another potential issue, if you were to use these drugs just for Alzheimer's in someone without HIV, is the theoretical risk of inducing resistance in latent retroviruses. That's something that would need careful thought.
Right, the safety profile needs careful consideration for a different patient population and potentially long-term use. Is research looking at ways around these issues, maybe developing related but safer compounds?
Absolutely. That's a very active area. Researchers are developing derivatives of NRTIs, like a compound called canine. The goal with these derivatives is to keep the beneficial inflammasome inhibiting effect, but eliminate the mitochondrial toxicity and the risk related to retroviral resistance. Canine is actually already in clinical trials, and impressively, in animal models of Alzheimer's, it showed it could completely reverse deficits in spatial memory and learning. So this represents a really exciting path forward, aiming for drugs that target the inflammasome effectively, but with a much cleaner safety profile suitable for chronic use in Alzheimer's prevention or treatment.
That's a very hopeful direction.
So just to wrap up with that practical summary again, this deep dive highlights research showing nucleoside reverse transcriptase inhibitors are associated with a significantly lower risk of Alzheimer's disease. The likely mechanism involves their ability to inhibit inflammasomes, pointing towards inflammation as a key therapeutic target. It's an important finding suggesting a potentially new avenue for tackling Alzheimer's.
It really makes you think. Considering these findings, the potential to repurpose existing drugs and the development of these newer, safer derivatives, how might this change how we approach Alzheimer's prevention or early treatment down the line? What are the implications for identifying people at high risk or for designing those crucial future trials that need to carefully weigh the benefits against the risks? It certainly opens up new questions.
It really does. This research nudges the door open further on inflammation as a central player. It encourages us to look critically at drugs we already have, maybe sitting on the shelf, for unexpected solutions to one of our biggest neurological challenges.
A fascinating area to watch. We strongly encourage you to read the full paper for all the details. Thank you for joining us for this weekly neurology deep dive, produced by America Vanini.